Yes, so my name is Chiu Fan Lee, and thank you very much for the opportunity for me to talk to you about my research. And so today's talk is on novel physics arising from phase transitions in biology. And so maybe I'll start by just summarizing a few phase transition examples in biology that are of interest in my group. So there are actually a few phase transitions phenomena found in biological systems in many different length scales. So the first example that I'm going to talk about is the formation of amyloid fibrils. So amyloids are intimately related to a lot of human diseases, such as the Alzheimer's disease, the Parkinson's, and type 2 diabetes. So these are nanoscale polymers. And so in many cases, we in our body have these endogenous proteins. They're in the normal form, the good form, but sometimes they can become misfolded, and subsequently these kind of biopolymers can form. So they're called amyloid fibrils. And the soluble protein to the misfolded polymeric aggregate or polymeric fibrillar form is a kind of a phase transition. Now, although strictly speaking, this is not a thermodynamic phase transitions, but this phenomenon certainly has all the salient features of phase transitions, such as a critical concentration beyond which amyloid fibrils can form. So that's one particular example. Now, moving up in length scale, we are now looking at in fact, an embryo 15 microns apart is an embryo of a roundworm, C. elegans, and it is the embryo right after fertilization is still in the one cell stage, and the green blobs in particular are called P granules, and they are important for the reproductive development of the organisms. And as you can see, there are green blobs everywhere at the beginning. Now, this movie is looped. So at the beginning, it is like this. So lots of green blobs everywhere. Subsequently, they all get localized to one side. And now we realize recently, relatively recently, since 10 years ago, that these P granules are formed via phase separation, very much like oil droplet formation in your salad dressing in any aqueous solution, for example. And in fact, I guess the realization that phase separation is playing such a major role in cytoplasmic and nucleoplasmic organization has, many believe, revolutionized cell biology. The way we understand how a cell organizes its interior. So this is the biomolecular condensase, P granules in this case, of course, a phase transition behavior. It's a phase separated drops rich in protein and RNA. Now, moving further up the length scale, now we are talking about millimeters, because we're looking at so-called wound healing assay. What we're looking at is basically a bunch of cells. Each cell is about 30 microns in diameter, approximately. And so this is called a wound healing assay because at the beginning of the experiments, the experimentalists grow a confluent layer of cells. And then as you saw, there's a void in the middle because they could scratch out the cells in the middle. And if you let it go, then the cells will just move inwards, and due to proliferation and movement, they subsequently just heal up that void, hence the name wound healing assay, akin to what happens if you cut your skin, for instance. So this is in physics, we call it active matter because these kind of material is very different from dead things because they can reproduce, they can move. So hence the name active matter. And one very interesting feature that for this active matter is that the whole system can move coherently. So i.e. every single, in this case, every single cell can be moving in the same direction as any other cells on average. And as a result, we can have something called the collective motion phase, where the whole system is moving in a particular direction on average. Of course, that system, thermal system, cannot display this kind of things because we are talking about a persistent current happening. And in active matter, matter, we can, because we're talking about active systems and biological systems where they are converting potentially chemical energies into this kind of mechanical energy. Okay, so these are some of the phase transition behavior we can see in biology, but in this short talk, I'm going to focus exclusively on the third topic, that is active matter. So for that, I'm going to kind of refine or narrow down the title a little bit. So I recall the title of the talk is Novel Physics Arising from Phase Transitions in Biology. So transitions, by phase transition, I meant the study of phases. And biology, I will narrow it down to a very specific attribute of biological systems, and that is active motility. So I can rewrite, I should redefine the topic to be novel phases in active systems, and that's what I will focus on from now on. Okay, so let's step back. If we are talking about phases, let's step back and think, what are phases after all, or states of matter? What are states of matter? Now, this is not actually that easy to define, but one crucial property is that distinct phases are always separated by phase transitions. So that's something. So by phase transitions, I meant thermodynamic discontinuities. So as a canonical example, when we talk about phases, obviously we start with a single component system, for example, a bunch of molecules that are interacting via the Lennard-Jones potential, for instance, and as you change the interaction strength or change the temperature, then you can get into different phases. And in particular, you can have a phase diagram here on the right. The parameters are temperature and pressure. You can be in the solid, liquid, or gas regime. Now, solid, liquid, or solid gas transitions are always separated by a phase transition. Hence, they are distinct phases by this definition, right? However, liquid gas transformation does not have to be separated by phase transition all the time. For instance, if I start here in the liquid regime, I can increase the pressure, increase the temperature, lower the pressure, and then lower the temperature, go to gas regime. So hence, liquid and gas are in fact not distinct phases, contrary to obviously to what most people learned in school, but that's just how it is. And we look at a slightly different property or a slightly different definition, and that's due to Landau, and say, well, distinct phases are characterized by distinct symmetries and conservation laws. So that's if we use that definition, then and go back to look at the gas, liquid, solid phase diagram again, we will arrive at the same conclusion. Liquid or gas and solid are distinct phases because they have distinct symmetries. In liquid, we have translational symmetry. In solid, we don't have translational symmetry because the translational symmetry is broken to produce a crystalline structure. However, liquid and gas remain the same phase because they have the same symmetry. So this is consistent with the previous property, the previous definition, where phases are just parameter regime, parameter domains that are separated by phase transitions. Okay, so now. Then let's ask this question. When a phase is a new phase, how do we know that? Let's say I study a new system, a biological system, a bunch of cells moving around. How do I know that what I'm looking at constitutes a new phase in physics? Now, if we use the Landau's definition of phases characterized by symmetries and conservation laws, then you may say, wow, you may write down all the symmetry, potentially new symmetry you found, and say, well, this is a new phase because it has new symmetries. But unfortunately, it's not easy because, in fact, the reverse of the definition is not true. Systems with distinct symmetries can belong to the same phase, can have the same hydrodynamic behavior. So even if you have new symmetry, you've identified new symmetries, doesn't mean that the system you're studying constitutes a new phase in physics. So what do we do? But then one solution is to use renormalization group method to identify novel phases. Now, renormalization group methods should be familiar. Well, at least the term should be familiar to all physicists. But basically, a RG method is a calculational tool that were invented by Wilson. Kenneth Wilson is a Nobel Prize winning work, and others in the 60s and 70s. And that calculational tool can allow us to elucidate the large distance or hydrodynamic behavior of the model systems, right? That's described by some model equations. So specifically, what we do is that we have input as the set of equation of motion that describe the system, and the equation of motions themselves are derived from conservation laws and symmetries. Now, I say equation of motions because I'm looking at non-equilibrium systems, typically non-equilibrium systems in biology. But if you are obviously for thermal system, then you would say you start with a Hamiltonian or a free energy that is constrained by the conservation laws and symmetries. So that would be the input. And then you start the calculations, which is basically a mechanical calculation, although the calculation itself can be very involved. But it's you turn the crank, and then your outcome is typically something called a RG flow diagram shown here. So the flow diagram is this is a cartoon with two axes. One is model parameter 1 and model parameter 2. And these model parameters are parameters in your model equations. So typically you would have many, infinitely, in fact, could be infinitely many model parameters. So here it is a sort of a collapsed view of the flow. And what the flow is showing is how these model parameters change as you do this RG transformation, which another way to say is a coarse graining operation. You start with a system where you know the microscopic details, and then you forget about the microscopic detail. You focus on the sort of larger scale behavior. And as you move to larger and larger scale, then the parameters change. And they go to a fixed point, as shown in this case. So the two model parameters go to a fixed point in this red circle here. Then the fixed point can tell us what phase the system is in. The fixed point is also called a universality class. Now, so this provides a way to identify novel phases, because if you identify a fixed point, and it's a fixed point that's new, you can compare to other, what the fixed points known in the literature, then you can conclude that the system that you are looking at with this type of symmetries and conservation laws constitute a new phase in physics. So let's look at go back to the input and use active fluids or active systems as an example. So the input here, again, are just conservation law. In this case, we have mass conservation and the symmetries. The symmetries are translational, rotational, temporal, chiral. These are very canonical, in the sense that not many people would debate about the validity of this symmetry. So for instance, translational symmetry, what it means is practically to say that if you were to do your experiments here in London, and and the result you get should be identical to someone doing the experiment somewhere else in, I don't know, in the US, right? Because it's translational invariance. It shouldn't depend on where you do it. That's translational symmetry, right? So these are very standard symmetries, and that's the only symmetries and conservation law that goes into the standard model of active fluids. And that's what Antona and Yu-Hai Chiu did in 1995. They started off with these symmetries. Conservation law arrived and then derived the generic equation of motion that describes active fluids. So we have mass conservation giving us a continuity equation. Rho is the density field and G is the momentum field. So this equation itself is constrained by mass conservation. And the momentum field equation, form of it is very complicated. Every term is constrained by the translational, the symmetries displayed up Okay. So another way to see it is kind of like a modified Navier-Stokes equation. In Navier-Stokes equation, we have two more symmetries. We have one more symmetry, got linear symmetry, and one more conservation law, which is momentum conservation, and that's it. So we delete those two things, we arrived at active fluids. And one particular feature of this active fluid, as I already mentioned at the beginning, is the fact that it can have a collective motion phase. So things in this active fluid can move together. So now we are kind of ready to answer partly the questions, right? Are there novel phases in active systems? The answer is yes. In particular, using the active fluid model, in the incompressible limit, the collective motion phase is a new phase, a new non-equilibrium state of matter when the spatial dimension is higher than 2. Now, however, if you are looking at 2-dimension active fluids, it actually is not a new phase. It belongs to the Carter-Parisi-Sen universality class, which is a well-known universality class that describes surface growth models. So this is one example where you can have systems with completely different microscopic physics, completely different symmetry, and yet they belong to the same universality class. Now, so we have one new phase, but that's not all. In fact, if we take the previous active fluid model to the other extreme, where you go to the infinitely compressible limit, i.e. you say the density field is completely decoupled from the momentum field, and because the momentum field doesn't care how compressed a local region is, then this model can again be solved using renormalization group method. And in this case, again, the collective motion phase is a new phase when the spatial dimension is higher than 2. So that is kind of, so that's echoes the title of the talk, novel phases in active systems. There are novel phases motivated by biological processes. Now, I would just like to, before I acknowledge the people involved, I would like to just give an outlook slides, and it's kind of like a research agenda in my group, right? So my group is a theoretical biophysics group in the Department of Bioengineering. But when we talk about biophysics, people tend to think of this loop here, this direction here. People use physics to study biology quantitatively, which is of course a very worthwhile endeavor. But what's less emphasized is the fact that biology can inspire new physics. The new physics here is the discovery of new phases by studying systems that are motivated, that are basically common in biology. So I think this is actually a circle where both fields can inspire each other. Now, I just want to advertise, take the occasion to advertise a fully funded PhD position to start in October. If you know people who are interested, then just ask them to go to findaphd.com and they would be able to find the position available. So, so this is my team at Imperial. The work that I actually mentioned was done in collaboration with Nerming Chen from the China University of Mining and Technology and John Toner from the University of Oregon. And thank you very much for your attention. Okay.
Thank you very much, Chiu Fan. So I haven't got any questions yet. If anybody does have questions, please either raise your hand or unmute and ask away. Okay, Rafi has a question.
Let me just unmute myself. Yeah, you were talking about renormalization group, and you mentioned symmetries, but if you want to use renormalization group, don't you also have to have scaling variance symmetry? And the question is,
no, you don't. You start with the equation of motion. We don't know. The renormalization group would be most useful if you can find a fixed point, but if the system does not display scale invariance, you may not get a fixed point. So you can always use the renormalization group, but it may not be, it may not lead you anywhere.
Well, if you don't have a fixed point or you don't have scale invariance, then renormalization group is not useful at all.
That's true. Well, you can still, well, it is less useful for sure. It may diverge, and then you may conclude something from that. You may say this is a first order phase transition due to fluctuation induced kind of modification.
This leads to my second question. Are the equations of motion that you presented, well, Navier-Stokes we know, are they renormalizable?
Well, this is a Wilsonian. Okay, what do you mean by renormalizable? It is, you can apply the renormalization group to get to a fixed point. It's a Wilsonian scheme. If you're thinking from the perspective of field theoretic renormalization, that doesn't really sort of feature in this particular condensed matter problem, because there's always an ultraviolet cutoff. So we never need to take the length scale to infinitely small.
Well, no, you mentioned the origin of renormalization group, Wilson renormalization. The idea is that if you have an equation of motion, if I take the scale, let's say in this case the spatial dimensions, and I multiply by lambda, let's assume that it's isotropic, okay? So I multiply by lambda to some power, then I get the same equations, maybe transformed by some power.
Until, that's true, from some ultraviolet cutoff to the thermodynamic limit, that's true. But you cannot do that when you make the length scale smaller than the ultraviolet cutoff, then you're pushing the...
Forget the cutoff, I'm not talking about the cutoff, you're right. If you have cutoffs, then it doesn't work. But my question is, are the equations, do they scale like that? In other words, can you use renormalization group, usefully in these equations that you presented?
The cutoff is crucial to the derivation, but the equation of motion, if you look at that, I wouldn't be able to tell whether it has a scale invariant structure in it until you do the renormalization group, and the renormalization group gives you a fixed point. Then I can say, well, when you go to the thermodynamic limit, there is a scale invariant structure, or basically it's the correlation. Time and space will follow some sort of power law. So that would be the conclusion coming from the fact that you have a fixed point in the RG, under the RG calculation. Yeah, so the equation, I guess a short answer to your question is, yes, the equation of motion has this scale invariant structures, but it will not be obvious from just looking at the equation without doing the RG calculations to begin with.
But you don't know, right? You haven't done this yet.
I have done that. I have done that with Leuming Chen and John Tongler. You can go to the two papers I've shown, and the calculations are there, so you can check it if you're interested.
Thank you.
Thank you.
Thank you, Rafi.